Thank you very much for the invitation, Philippe and Brett. It's been a great meet. It's a great city, but it's also been a great meeting. So I think to my topic, I've got a great introduction in the first talk by Alan Walker, and again this morning by Gérard Hébert. We will stick to neonates now, to this window of opportunity. And in difference to Gérard, I would like to try to convince you that maybe the epithelial cells are the chef d'orchestre key, kind of, sit on top of everything and kind of make the difference. How do I do this? Right. Okay, just some words about epithelial cells. I don't think I need to introduce the way mammalian cells are able to recognize bacteria, just to mention that epithelial cells, similar to some professional immune cells, maybe to somewhat restricted spectrum, but they do express pattern recognition receptors of different flavors, like TLRs, not like receptors, inflammasome proteins, or even helicases. That's the one thing. The other thing is that the epithelium in the gut is really very complicated, and I'd like to mention here that I will focus on the small intestine, which is very different from the colon. Nevertheless, in both organs, we have a continuous migration. We have stem cells to proliferate, and then you get a continuous migration and differentiation, which is important to keep in mind. And the other issue is that if I talk about epithelial cells, I actually mean 5 different types of epithelial cells, which are stem cells in the crypts, close by the Panna cells, the enteroabsorptive enterocytes, the majority, but also goblet cells that Morlin has mentioned yesterday, and enterochromaffin cells. And Nita has talked about the Panna cells very much. All right, I think we all know now that innate recognition at the epithelial barrier of microbes can be beneficial. And in one sense, that's very easy to grasp. That's about infection. You want to recognize the infecting agent to defend yourself and kind of clear the infection. But there seems to be more about it. It seems to be that even for homeostasis, you want to recognize the microbial flora. So I think that's quite firm evidence there for that. But there's also firm evidence that too much recognition, obviously, is not good. So you need to control this. You need to kind of balance the way you recognize the microbial flora in the quantitative manner. So just because I will focus, at least in the first part, on TLRs, just a short introduction, I don't think I need to say much about it. Just to say that there are a number of those receptors and they're signaled via conserved pathways, which means that if you would interfere with a given molecule involved in this pathway, you would interfere with signaling of all of the TLRs. And just to mention one molecule I will talk about later is a small kinase involved in the MYD88 dependent TLR pathway, which is IRAC1 involved here. Right, so the work started actually approximately 10 years ago when I still did my postdoc at the Karolinska with Stefan Normack, and we've got hold of a cell line, small intestinal mouse cell line, from a colleague here from Paris, Alain Vondeval, and we noticed, and at the time there was a big surprise for me, that these cells responded very nicely to LPS and produced lots of the chemokine called MIP2. They did make RNA for TLR4 and MD2, and they did make the protein for TOL4. And this was kind of different from what at the time people thought that, the epithelial cells would not be able to see microbes because, for obvious reasons, they would induce inflammation, it wouldn't be good. But this type of responsiveness of these cell line in that sense made us, trying to understand, is that really true in vivo, and what is maybe the underlying mechanism that we still live healthy without having inflammation despite having the microflora in the gut. So Michael Lotz, a PhD student in my lab, addressed the question then in vivo. So he did laser microdissection of the epithelium to look for TOL4 expression. He isolated gut epithelial cells, so this is a villus, and you have the attached crypts here. They're still polarized, and did a FACS scan for TOL4 expression. He could say that, yes, seems to be RNA and protein for TOL4. Then he tried to cultivate those and stimulate, and this is very tricky because they're not really, they don't really like to be cultured once they've lost the contact to the basal membrane. But what he could find, it was a surprising finding, that if he took epithelial cells from fetal mice, they did produce some amount of chemokine response to LPS, whereas cells they took from mice postnatally did not. Okay, so we thought about this, and then we thought maybe that's an adaptive thing that, around birth you want to shut off your ability to recognize the endotoxin. But if that would be the case, this would reminded us to something that's known for a long time in macrophages. That's called endotoxin tolerance, meaning that if you expose a macrophage to LPS once, the macrophage response. But if you come a second time with the endotoxin the day after, these cells would not respond any longer. No, but this would implicate that you would have to see a little stimulatory peak between the fetal and the postnatal situation. So we went on looking for this, and that was actually quite tough because, as you know, mice give birth at night, normally something like 5 o'clock in the morning, and we wanted to really carefully look whether we can see any kind of activation of the epithelium. So this is a time kinetic hours after birth. So this is 5 o'clock in the morning, 6 o'clock in the morning. See this? Okay, and we isolated the gut epithelial cells and looked for transcriptional activation. And indeed, what we could detect is after vaginal delivery, 2 to 4 hours, there's a little transcriptional activation in the epithelium. You also can see P65 translocation and IKB alpha phosphorylation in normally born mice. They're not exposed, they're not infected, nothing. Okay, and then Michael discovered that with this activation, he saw degradation, disappearance of this little kinase ARAG1 in the isolated epithelium after birth. And that was interesting because ARAG1 is required for signaling via TLR4. So we thought, okay, so maybe this is the mechanism of tolerance in these gut epithelial cells. And at this stage, there came a postdoc in my lab, Cécilia Chassin, a very talented postdoc. She spent the last 3 weeks in my lab. It's just got a position here in Paris 2 weeks ago, I think. And she will be back in Paris in a couple of months and then continue her work here. And she kind of was then very interested in, so what's the role of this ARAG1 and how is the ARAG1 disappearance regulated in the gut epithelium? Okay, so for example, the first thing she did a long kinetic and saw that ARAG1 protein comes back after weaning. So there seems to be a time window where ARAG1 proteins is degraded or very much reduced. There's no difference in mRNA. And she developed a new technique to test the ability of the cells to respond to LPS, and she could really correlate this disappearance of ARAG1 to responsiveness. So fetal cells did respond, postnatal cells didn't. After weaning, they responded to LPS again. Also, if you removed ARAG1 from epithelial cells using ARAG1 knockout mice, they didn't respond. So ARAG1 really seems to be essential for epithelial cells to respond to LPS. And if you remove ARAG1, that's completely sufficient to confer tolerance. Now, how to study the importance of this? That's difficult because either you would have ARAG1, then you can respond, or if you would remove this, then there's no way to then stimulate the cells. However, we made one observation that helped us very much, and that is that this transcriptional activation of the epithelium shortly after birth is only observed if you look at mice that are born via the vaginal delivery. So those are the white bars here. So they make this transcriptional activation. It's just transient. If you look at mice that are born by C-section or mice that lack TLR4, they don't do this activation and they maintain expression of ARAG1 in the epithelium. So we cannot use TLR4 knockouts to test the endotoxin effect, but we could use the C-section mice now to test whether the persistent expression of ARAG1 would make any harm to those mice. Okay, so we exposed C-section born mice with E. coli, just for the simplicity. And what we detected was a massive induction of epithelial apoptosis, here shown by tunnel staining and caspase 3 staining. And you can also use actin cleavage, which in epithelial cells is quite specific for apoptosis. So it's the full-length actin, beta-actin, is cleaved into 2 fragments, depending on what antibody you're using. Right, so importantly, if you use vaginal delivered mice that do not express ARAG1, expose them to E. coli, there will be no actin cleavage and no apoptosis. If you use C-section born mice, expose them to E. coli, you see actin cleavage, you see apoptosis. If you use TLR4 knockout mice, you will not. So it's a TLR4-driven, endotoxin-driven, TLR4-driven process of apoptosis. Okay, so how's the downregulation of ARAG1 on the molecular basis? How is that done? It's known in macrophages that ARAG1 is recruited to mediate, is then actually phosphorylated by ARAG4, is autophosphorylated very much, then binds to TAF6, and then ubiquitinated and actually degraded via the proteasome. Okay, so we tested this in these epithelial cells in comparison with the macrophages. We used inhibitors of the proteasome, but also of the lysosome. Okay, so in macrophages, inhibition of the proteasome inhibits degradation of ARAG1. Not so much if you inhibit the lysosome. In epithelial cells, it seems to be more complicated, the ARAG1 degradation, because inhibition of the proteasome as well as inhibition of lysosomal enzymes did impair the degradation of ARAG1. So this is protein level of ARAG1. Now this is responsiveness, and that confirms completely this observation that the macrophage is just inhibition by the proteasome inhibitor, enhances the production of the chemokine to LPS. It's actually prolonging the answer, whereas in epithelial cells, also inhibition of the lysosome does their job. Now, is that important in vivo? So if you give mice, vaginal delivered mice that normally do not have much ARAG1, proteasomal lysosomal inhibitors, you get ARAG1 back. If you expose those mice now to E. coli, they cleave actin, and they do get apoptosis of the epithelium. So it seems like, indeed, yes, ARAG1 needs to be degraded by the proteasomal lysosomal pathway to protect the mice from apoptosis of the epithelium. Okay, and then there came a paper from the Baltimore group, and what they proposed is that ARAG1 is also controlled by a microRNA 146A. Now, microRNAs are non-coding small RNAs that are processed, they form a complex with certain proteins, and then can bind with high specificity to certain mRNAs, and then lead to degradation of the mRNA. Or if they don't bind that tight, that's the idea, they are able to just induce translational repressions. So the RNA wouldn't be affected, but protein wouldn't be made. So in our case, we didn't see any difference in the RNA levels, so we thought maybe this is what we should look for. The interesting thing is the Baltimore group published that it should be degradation of the ARAG1 mRNA. So we got kind of been a bit careful, but thought, okay, so how to address this? We're just going to look whether the microRNA 146A is actually induced in the epithelium. And that was very convincing, the result. So those are isolated epithelial cells, time kinetic after birth. This is with birth, you see a strong increase of the microRNA 146A expression, and then there has a high expression during the first 2 weeks after birth. And then it gets down again. What's interesting here is, again, this is TLR4 dependent. It seems to be also birth mode of delivery dependent, so it's very similar to the ARAG1 degradation, and also the time kinetics is very similar. So you have this window of 2 weeks where you have high microRNA, precisely the window of where we have no ARAG1 and very low responsiveness. Okay, so is microRNA functionally important in epithelial cells? Now, in vitro, we could show that given anti-microRNA 146A would replenish ARAG1 protein, and would also enable the cells again, even though they've been tolerized, to respond to LPS and translocate B65 to the nucleus. So it seems to be, yes, in epithelial cells, microRNA 146A is important. What about in vivo? Okay, I told you that we have like 3 situations here. We have the vaginal delivered mice, high microRNA, C-section bone mice, low microRNA. Now, if we give vaginal delivered mice that have high microRNA, anti-microRNA, if we antagonize the microRNA, we can do this. It's taken up by the epithelial cells quite efficiently. The level of microRNA is diminished. Now, those mice re-express ARAG1 protein, and they get susceptible now to epithelial apoptosis. We can also do the opposite. We can take C-section bone mice that have low levels of microRNA. Now we give mimic microRNA, pretty much the same. It's just now not the antagonist, but the agonist. It will be taken up by the epithelial cells after birth. We get huge amounts of microRNA expression. ARAG1 protein is diminished, and these mice get protected from E. coli-induced epithelial apoptosis. Okay, so we kind of left with this type of model. We get TLR signaling. The signaling induces ARAG1 phosphorylation, ubiquitination, and degradation. And the degradation is required to diminish the protein levels. At the same time, the signaling induces NF-copper-B, which induces microRNA 146A, which leads to translational repression of protein expression of ARAG1. So the levels of protein, ARAG1 protein, are very much diminished by the 2 mechanisms simultaneously. That makes lots of sense if you think about an acute stimulation of the cells. However, there were some observations that really didn't make us thinking more. One was, for example, in the mixed cells, in the cell line, that if you took cells and tolerized them, ARAG1 disappeared. Now, if you wait for 2 days or so, if you expose the cells then to proteasomal lysosomal inhibitors, ARAG1 came back after 3 days. The other experiment was if you tolerize mixed cells again, they didn't, and then you wait a bit, the chemokine production goes down to baseline. But if you now add proteasomal lysosomal inhibitors, they spontaneously kind of shoot and make chemokine, high levels of chemokine production, and this chemokine production is TLR4 dependent. And the last observation was that as I told you before, if you give LPS, ARAG1 is downregulated, but if you, after the tolerization, so after 2 days, add as iRNA for TLR4, so reduce the protein expression of TOL4, ARAG1 comes back. And the only explanation for all these 3 findings is that actually the tolerant state, the fact that these epithelial cells, days after tolerization, remain tolerant, requires the continuous degradation of ARAG1, requires somewhere TLR4 protein expression to maintain tolerance. So tolerance isn't a passive state. It's not like a tolerant cell sits there and is kind of paralyzed, not responding, but rather a tolerant cell is running this process all the time. It's all the time degrading ARAG1 by phosphorylation and by translational repression. But that would implicate that they need the ligand, right? Because it's TLR4 dependent. And that actually is something that's known for a long time, that many cell types, also epithelial cells, and we've also shown this, they internalize LPS and keep it for a long while and are able to signal from this internal side. Okay, so, but this is all in vitro. What about in vivo? Okay, if you take from a 10-day-old mouse epithelial cells, this is the level of ARAG1 you would see. Now, if these cells have been exposed to proteasomal lysosome inhibitors, ARAG1 indeed comes back. You can also then look at the responsiveness, and again, the proteasomal lysosomal inhibitors lead to a spontaneous upregulation of chemokine mRNA. Okay, we thought, now that's interesting. Now, if tolerance is active state, what about this? Remember the long, sustained elevated microRNA expression. Maybe this is the reason why we have so long. This duration is so long. So we took ICs from neonatal mice and then exposed these mice to as RNA for TOL4 ARAG1. And now microRNA is really reduced, which indicates that indeed the sustained levels of microRNA are due to the fact that the epithelial cells in the gut of the neonate mice are continuously signaling via TOL4 and ARAG1. Okay, so this is the microRNA. What about coding genes? I mean, if you express microRNA, why not expressing real coding genes that make protein? So we took, again, the epithelial cells, looked into the situation shortly after LPS. Now, this is 6 hours after LPS. We call this ARAG1 high conditions because they would have lots of ARAG1 protein. We get the usual suspects, the usual chemokine genes, TLR2 very regulated in mice, and so on. Many other genes are down. However, if you wait now for 3 days, 96 hours, these mice have been the cells have been exposed to LPS and then watched and then left for 96 hours without LPS. We see that these genes are now down, not so unexpected. But we see other genes being now highly upregulated. And now these genes, in contrast to the first group of genes that are associated with inflammation, are associated with metabolism, cell differentiation, cell survival, cell cycle control, and metabolism. Okay, so we think that the tolerant state isn't just a passive state of non-responsiveness to a TLR ligand, but rather is actually an active state that drives cell differentiation. We think that this is important in the neonate gut. But we still left with one question, which is that why then ARAG1 comes back at weaning? Why is this process stopped around weaning and ARAG1 comes back? Okay, there is an old observation that many people have made when they use cell culture and might have used it, LPS-contaminated serum, is that if you change to pure media, to pure serum that is endotoxin-free, and just wait a bit, your cells get responsive again. And this is exactly the case. So if you tolerize cells and just wait, just propagate them, they will regain susceptibility. And what's interesting, if you now look at this course, it takes something like 7 days, splitting every second day. The endotoxin concentration in the epithelial cells during this time drops. microRNA drops, and ARAG1 protein comes back. This does not happen if you don't propagate the cells. So if you just leave them sitting, just change the medium, but don't propagate them, they don't regain responsiveness, endotoxin concentrations remain, microRNA remains high, no ARAG1 protein. Okay, so this is the cell line. That's actually known. But what about in vivo? And there's an interesting thing. I showed you in the beginning this scheme of the small intestine, villus and crypt. Now, this is actually wrong for the neonate, because neonate mice do not really have crypts. They have something that's called intervillus junction. So you have something like a pre-stem cell in the middle, but not really crypts. There's no massive stem cell. There's no massive cell proliferation, actually. It's pretty stable. The cells that you see at day 1 will be pretty much the same cells that you see at day 10. All right, so what we thought, maybe it's something to do with the emergence of this proliferation, which happens around weaning. Okay, so if you give LPS, endotoxin that's been labeled to die at 1 day after birth, and look, what about LPS in the epithelium? You see that, now this is the control, untreated. This is 6 hours after birth. You see, yeah, the epithelial cells have taken up the LPS. Day 3, they still have it. Day 6, they still have it. Day 14, they seem to lose it. It's getting much less now. Day 21, it's lost. And the persistence of the endotoxin in the gut epithelium goes along with the stable situation in the neonate with very little proliferation. So this is Ki67, staining. And with the emergence of high stem cell proliferation around weaning, we lose the endotoxin. That's not because the cells proliferate so much, because the cells are renewed. So the endotoxin positive cells are quite kind of shed into the lumen and kind of lost. And the newly produced cells are endotoxin free. Okay, so this is the model we're left with. We have the neonate, we have the fetal, high ARAG1, low micro. And then at birth, we get induction of microRNA, we get degradation of ARAG1 through endotoxin signaling. That's the neonatal phase. Interestingly, that's the period when we get colonization. And it's interesting to note maybe that depending on the neonate, depending on what environment the neonate is exposed to, it's being colonized by different types of flora. So the epithelium seems to be somewhat refractory to respond. But in the adult mouse, the situation has changed. After weaning, ARAG1 comes back as a protein, microRNA is low. I think that's not complete. Morlin would add here the mucus. Things get more complicated, and we think that now in the adult situation, for some reason, the cells can be responsive and fulfill a function there without being stimulated all the time. And I would like to make a connection here to a disease that has been mentioned before by Alan Walker, and which is necrotizing enterocolitis of the newborn. That's a disease that's quite frequently seen in premature humans, and the disease has been associated, what I said, with prematurity, but also with the colonization, particularly with gram-negative bacteria. And histologically, you see massive apoptosis. And in animal models of neck, there has been a clear role of TLR4 being identified. Okay, so we think that the gut epithelial cells, there is this maybe window of opportunity, but also a very special window after birth that confers some type of tolerance, non-responsiveness, non-exaggerated responsiveness, at the same time a permanent signal that drives differentiation. Just to mention one story to illuminate that this is not a general phenomenon. It's not like the epithelial cells in the neonate gut are completely unresponsive. They are unresponsive in the sense that they don't make pro-inflammatory mediators in response to infection. However, they do respond, of course, to other things. And this is a story that has been worked up by Johanna Pott, who is a PhD student in my lab, and deals with rotavirus. And I'm sure you know that rotavirus actually affects neonates. This is true for mice. That's actually also true for humans. Humans get infected up to the age of 6, roughly. What's interesting about rotavirus, and that's a staining of the small intestine, the red staining is the rotavirus, it is has a high tropism for the epithelium. And you can see this here. So it will infect gut epithelial cells very efficiently. It's a non-enveloped, as most of the enteropathogenic viruses. Segmented double-strand RNA virus has the strong epithelial tropism. It is inducing a diarrhea in infants up to 5 years of age. Actually, worldwide, rotavirus has a very severe impact. There's a high mortality associated with rotavirus, particularly simply due to the lack of access to medical care. Okay, that's a bit crowded, I know. The point I want to make here is rotavirus is recognized by helicases, Rig iron MDF5, as well as maybe TLR3. Now this induces activation of the cell and in turn production of interferons. Now interferons are then in turn recognized by the interferon receptor and then induce the expression of interferon response genes, many of which are antiviral. Okay, so the interesting thing here is that there are 2 types of interferons that have been associated with antiviral protection. One is type 1 interferon alpha and beta, and one is type 3 interferon, which is interferon lambda. Now, these have different receptors, but they signal exactly the same pathway. And people have, for a long time, been thinking, so what exactly is the contribution of lambda interferon to the host response, to the interferon response? Now, for example, people have used lung viruses, many different types of lung infection by viruses, and looked at different knockouts, like the type 3 interferon knockout, which is the IL-28 receptor alpha knockout, IFNA knockout, and the double knockout. Now, see here, the type 3 interferon knockout didn't really give any phenotype, at least not in the single knockout. Just if in the absence of IFNA, the type 1 interferon receptor, you saw a little difference. But as on its own, it didn't seem to have any effect. So what's then the role of this type 3 interferon? So there was an interesting finding by a group in Braunschweig, which showed that if you induce, that they had a reporter mouse here, that responds to interferon. Now, if you inject interferon beta, you get a strong response in the liver, particularly. But if you inject interferon lambda, what you get is a response, particularly in the gut and in the lung, so in the epithelial organs. And they could confirm this by staining for MX. So those are transgenic mice that make the MX1 protein. And they could show that now, indeed, the epithelial cells seem to somewhat respond to this interferon lambda. Okay, so we thought, if rotavirus is so trophic for epithelial cells, let's try this model with these animals. So we used wild type 3 interferon knockout, receptor knockout, type 1 interferon receptor knockout, and the double knockouts. Infected with rotavirus and looked for virus replication. And now the first time we could see, indeed, in the single knockout for type 3 interferon, a strong enhancement of viral replication. Pretty much the same as the double knockout. Not much of a difference. You can also see this if you look at the virus protein production. And now, what's the reason for this particular phenotype? And that's nicely seen if you look at the single cell level. Now, in green, we have MX protein. That's an interferon response gene that is antiviral. In red, it's just ecatrin to stain the epithelium. In blue, DAPI, and in white is rotavirus. Now, the wild type situation, neonate mouse infection, we have little rotavirus replication, but very nice MX protein expression in the epithelium. It's probably protective. Now, if you look at the single IL-28 receptor knockout mouse, we get lots of rotavirus replication, lots of white cells, almost no MX protein in the epithelium, but we still have MX protein in the lamina propria, but that doesn't seem to help the mouse. If you look at the IFNA mouse, we have little rotavirus, nice MX protein in the epithelium, no MX in the lamina propria, and the double knockout mouse is kind of the same. But you don't have any MX response. And you have, so that's the same like the IL-28 receptor knockout. Right. So it seems like there is a cell type specificity of the two types of interferon. Now, if that's true, we should be able to protect mice from rotavirus by just injecting lambda interferon, better than by injecting alpha interferon. So this is what we did. We exposed mice to orally to rotavirus, and injected interferon. So either interferon alpha or interferon lambda. And indeed, the single dose, treatment of the lambda interferon protected the mice, and they shed less virus as compared to treating the mice with type 1 interferon. So here we left with a model that the epithelium is specifically, now this is the wild type having type 1 and 3 interferon receptors. Now here the epithelium would respond and the lamina propria cells respond. The mouse is protected to a large degree because, the epithelial cells do respond. Now, if you remove the type 3 interferon receptor, the epithelial cells are unable to respond to this viral infection, and you get lots of virus replication. It doesn't help that the lamina propria cells are able to respond, because in the inverse situation where the lamina propria cells are not able to respond, but the epithelial cells are, you have low virus replication. So the interferon, the type 3 interferon role is a cell-specific role to activate epithelial cells to respond to rotavirus infection. And with that, I would like to finish and just mention again the people that did the work. Michael Latz, who left the lab, did the first part, then Cecilia Chassin, a French postdoc who will start here now her own group, and Johanna Pott, who just finished her PhD. Thank you very much.